Hello everyone, today let us discuss about DNA. First, let's come to introduction. Deoxyribonucleic acid is an important molecule that carries genetic information for growth, development, functioning, and reproduction for all living organisms and many viruses. Understanding of DNA is concerned with understanding how genes control the chemical processes within cells. Most of the DNA is in the classic Watson-Crick model, simply called bDNA or B-form DNA. Apart from it, many different forms of DNA are present. Study of nucleic acids and nucleotides require proper description of base sequences. There are various rules for proper representation of repeating units of nucleotide residues, without which the description will be cumbersome and difficult to interpret. Today, let us discuss on the structural diversity of the DNA molecules and notation for describing polynucleotide chains. Before discussing the different forms of DNA, it is necessary to understand the double helical structure of DNA. So now let us come to the double helix. Discovery of the double helical structure of DNA by Watson and Crick was an important event in molecular biology. Their proposal was the outcome of synthesis of many significant discoveries. One important clue to the structure of DNA came from the work of Irwin Sargraf and his colleagues in the 1940s, which were presented as Sargraf rules. One of the findings is that the number of adenosine residues is equal to the number of thymidine residues, and the number of guanosine residues is equal to the number of cytidine residues. Another landmark discovery was in 1950s, when Rosalind Franklin and Maurice Wilkins observed a characteristic X-ray diffraction pattern of the DNA. From this pattern, it was deduced that DNA molecules are helical with two periodicities along their long axis. In 1953, Watson and Crick postulated a three-dimensional model of DNA that could account for all the available data. The Watson and Crick model of DNA proposed that the two right-handed polynucleotide chains are coiled in a helical fashion around the same axis. The polynucleotide chains are formed by deoxyribose sugar and phosphate linkages. Each deoxyribose sugar is linked with nitrogenous base, either purine or pyrimidine, at its carbon number 1 position. Purine may be adenine or guanine, while pyrimidine may be cytosine or thymine. The two sugar-phosphate backbones are on the outside of the double helix, while the bases project into the interior. The adjoining bases in each strand stack on top of one another in parallel planes. The orientation of the two strands is different, that is antiparallel. The two strands of the double helix structure are held by formation of hydrogen bonds between purine and pyrimidine bases of the respective linear molecules. The pairing between purine and pyrimidine nucleotides are very specific. In a double helical structure, adenine in one strand is paired with thymine in opposite strand through two hydrogen bonds. Guanine is paired with cytosine through 3 hydrogen bonds. It provides the rationale for Sargraf rule that in any DNA, adenine is equal to thymine and guanine is equal to cytosine. The offset pairing of two strands creates a major group and minor groups on the surface. Now let us come to types of DNA. Model described by Watson-Crick is referred to as B form of DNA, which is normal form present in most DNA molecules in the cells. It is to be noted that DNA is a flexible molecule. Rotation is possible around a number of bonds in the sugar-phosphate backbone. Moreover, thermal fluctuation can produce bending, stretching, and unpairing of the strands. Many important deviations are found in cellular DNA, some of which play important roles in DNA metabolism. In addition to measure B form, measured DNA structures are A form and Z DNA. Now let us come to B form of DNA. It is the model originally proposed by Watson and Crick. It is the most stable structure for a random sequence DNA molecule at neutral pH and physiological salt concentration. It is also considered as the standard point of reference in any study of DNA. In this model, the two strands are arranged in right-handed manner. It is 23.7 angstrom wide and extends 34 angstrom per 10 base pair of sequence. The helix makes one complete turn about its axis for every 10.4 to 10.5 base pairs. The major group is wider than the minor group in bDNA, and many sequence-specific proteins interact in the major group. Now let us come to A form of DNA. A form is the alternative model with thicker right-handed duplex with a shorter distance between the base pairs. It has a shallow, wide minor group and a narrower, deeper major group. The A form occurs under non-physiological conditions in partly dehydrated samples of DNA. In the cell, it may be produced in hybrid pairings of DNA and RNA strands and in enzyme DNA complex. In this form, DNA is arranged in right-handed double helix. However, the helix is wider and number of base pairs per helical turn is 11 as compared to 10.5 in bDNA. The major difference between A form and B form nucleic acid is in the conformation of the deoxyribose sugar ring. It is in the carbon number 2 endo conformation for B form, whereas it is in the carbon number 3 endo conformation in A form. Now let us come to zDNA. zDNA is widely different from the B structure by the presence of left-handed helical rotation. In this model, the double helix winds to the left in a zigzag pattern. The zDNA helix has a structure that repeats every 2 base pairs. zDNA can form when the DNA is in alternating purine-pyrimidine sequence such as G, C, G, C, GC and G and C nucleotides in different conformations leading to the zigzag pattern. zDNA is commonly believed to provide torsional strand relief, that is supercoiling, while DNA transcription occurs. There are 12 base pairs per helical turn as opposed to 10.5 in bDNA. The zDNA appears more slender and elongated as compared to native DNA. Major group is barely apparent in zDNA and the minor group is narrow and deep. Now let us come to the comparison of aDNA, bDNA and zDNA. Helix ends. aDNA has right-handed helix ends, bDNA has right-handed helix ends, and zDNA has left-handed helix ends. Second parameter is residues per turn. aDNA has 11 residues per turn, bDNA has 10.5 residues per turn, and zDNA has 12 residues per turn. Next parameter is axial rise in angstrom. aDNA has 2.55 angstrom axial rise, bDNA has 3.4 angstrom axial rise, and zDNA has 3.7 angstrom helical rise. Next parameter is helical pitch in degree. aDNA has 28 degree, B 34 and Z 45. Base pair tilt in degree. aDNA has 20 degree tilt, bDNA has -6 degree and zDNA has 7 degree tilt. Next is rotation per residue in degree. aDNA has 33 rotation per residue, B has 36 and Z has -30. Next parameter is diameter of the helix in angstrom. A has 23 Armstrong, B 20, and Z 18. Next parameter is glycosidic bond configuration. For deoxyadenine, deoxythymine, deoxycytosine, for A DNA it is NT, for B DNA it is NT, and for Z DNA NT, for deoxyguanine has NT, NT, and syn. Next parameter is sugar pucker. For A DNA is C3' endo, for B DNA it is also C2' endo, for Z DNA C2' endo, while deoxyguanine has C3' endo, C2' endo, C3' endo. Next parameter is intra-strand phosphate-phosphate distance in Armstrong. In this, A DNA has 5.9 Armstrong, B 7, and Z 7, while deoxyguanine has 5.9 Armstrong, 7, it is 5.9 Armstrong. Next parameter is topology of measure group. For A DNA, it is narrow and deep, for B, wide and deep, and for Z, it is flat. Next parameter is topology of minor group. For A DNA, it is broad and shallow, for B, it is narrow and shallow, and for Z, it is narrow and deep. Along with these forms, there are other forms of DNA. In short, the double-stranded DNA exists in at least 6 forms, that is A DNA, B DNA, C DNA, D DNA, E DNA, and Z DNA. Triple-stranded DNA. Popular model of DNA is double-stranded. However, there is also triple-stranded DNA. In this model, 3 oligonucleotides wind around each other and form a triple helix. One strand binds to a B-form DNA double helix through Hookesten or reverse Hookesten hydrogen bonds. Now let us come to single-stranded DNA. Almost all the organisms contain double-stranded DNA except a few viruses such as bacteriophage 5×174, which consists of single-stranded circular DNA. It becomes double-stranded only at the time of replication. Now let us come to circular DNA. Biological DNA in higher organisms exists as long linear double helices. However, almost in all the prokaryotes and a few viruses, the DNA is organized in the form of a closed circle. The two ends of the double helix get covalently sealed to form a closed circle. Now let us come to tetraplex DNA. 4 DNA strands can also pair to form a tetraplex or quadruplex. It occurs only for DNA sequences with a very high proportion of guanosine residues. They are normally located near the ends of the chromosome or the better known as telomeric regions and in transcriptional regulatory regions of multiple oncogene. On the basis of location, DNA can be divided into 2 major types of DNA, the genomic DNA and mitochondrial DNA. Genomic DNA or nuclear DNA, it is present in the nuclear region. In the eukaryote, they form part of chromosomes. This type of DNA comprises the genome of an organism. It controls expression of various traits in an organism. Now let us come to mitochondrial DNA. It is present in the mitochondria. Mitochondrial DNA are believed to derive from circular bacterial genomes. It is double-stranded circular molecule. It is maternally inherited. It does not change from parent to offspring. Chloroplasts also have their own DNA, often abbreviated as cpDNA. It is also called plastome. Now let us come to notation for nucleotides. In the polynucleotide chain, there are repeating units of phosphate, sugar with radiating bases. While describing structures or reaction mechanisms of the nucleotides, it would be cumbersome to always show the respective phosphodiester and sugar molecules. So presentation of this repeating unit by shorthand notation has become a practical approach. Representation of the initial letters or bases became an accepted method for abbreviation. Another analogous procedure was adopted for the other nucleotide components is also adopted. The notation currently in use was first formalized by the International Union of Pure and Applied Chemistry in 1970. Under the commonly used IUPAC system, bases are represented by the first letters of their chemical names. Guanine by G, cytosine by C, adenine by A, and thymine by T. This shorthand also includes 11 ambiguity characters associated with every possible combination of the 4 DNA bases. Now let us come to the trivial names of nucleosides. Trivial names of nucleosides are derived from the names of bases by replacing their endings. Purine and pyrimidine nucleosides are formed by replacing the suffix -ocene and -idine respectively. Ribonucleosides, their respective abbreviations and nucleoside symbol. Adenosine, abbreviation ADO, symbol A. Guanosine, abbreviation GUO, symbol G. Ionosine or base hypoxanthine, abbreviated as INO and symbol as I. Xanthosine, XAOX. Cytidine, CTD, symbol C. Uridine, URD, symbol U. Ribothymidine, THD, symbol T. Deoxyribonucleosides, deoxyadenosine, DADO, DA. Deoxyguanosine, DGUO, DG. Deoxycytidine, DCTD, abbreviated as DC. Thymidine, DTHD, abbreviated as DT. Nucleotides are usually named as the phosphates of their nucleosides. In the nucleotide, numerical locand 5' may be omitted. In the nucleoside 3' or 2' phosphate, the locand for phosphate must be given. In one representation, the letters are joined by a zigzag line. Horizontal line symbolizes sugar and diagonal line indicate the way in which they are linked. When the diagonal line meets the end of the horizontal line, it indicates a 5' linkage, whereas when it meets the middle of the horizontal line, it indicates a 3' linkage. In another representation, the lines are eliminated altogether and the formula is simply written in the form PA, PC, PG, and so on. In the nucleotide, letter P is used to indicate a phosphate group. In this notation, when P comes before a letter, it indicates that the phosphate is attached to the nucleoside by a 5' linkage. In the most abbreviated nomenclature, PN stands for 5' nucleotide, NP for a 3' nucleotide, and DNP for 3' deoxynucleotide. This notation is based on the convention that an oligonucleotide chain is drawn horizontally with its 5' hydroxyl group is at the left and its 3' hydroxyl group at the right hand end. The poly A means multiple A's at the 3' end. More common method representation of nucleotide sequence is to omit the P and write only the order of bases such as GGCTA. This notation assumes the presence of phosphodiesters joining adjacent nucleotides. DNA sequences are usually preceded by a lowercase d to denote deoxy, as in DGGCTA. So finally, let us come to conclusion. DNA is the chemical basis of heredity and fundamental units of genetic information. Knowledge of the structure and function of nucleic acids is essential in understanding the transmission of hereditary characters. It is also important for managing pathophysiology, as well as the genetic basis of disease. For proper understanding, it is essential to have proper knowledge of shorthand notations for representing accurate data. Deviations of DNA structures and fluctuations in DNA structures according to changing cellular conditions.